today I'll be talking about design, construction, and usage of the draft pangenome. And so this will actually be mostly about other people's research. If you want to see my research, this talk actually tees it up for my research. So you have to go back in time and watch it on YouTube for my Tuesday talk. Okay, so credit to the consortium for the Human Pangenome Reference Consortium, who did a lot of the work that I'm presenting here. And here is just a photo of the last meeting last fall, right before the first draft pangenome was published. Okay, so let's first talk a little bit about history. So when the first human genome was sequenced, so first I was talking with John about this. How many people here were born after the first release of the human genome? Okay, very few, but maybe I'll move to, I won't ask how many people were in grade school. Okay, so there's this great cover which shows a bunch of different humans in the genome, or different humans, and it's this one genome that kind of all brings us together. But let's actually kind of dive in a little bit into the methods that were used to build the pangenome. Genomic DNA was taken, sheared into fairly large 240 kilobase fragments to build what's called a back library. So these are ways of amplifying really long segments of DNA. And each of these segments was then sequenced and merged into a build of the human assembly, such that every back sequence represents the sequence of a single individual. Okay, in this case, there were 7 different samples that were used to build the human genome, but if you look at the base at any one position, it represents exactly one individual's DNA, and even just a haploid part of one individual's DNA. And so what is that missing? Okay, that's missing a lot of diversity that we know about, okay? So this is a view from the 1,000 Genomes project. We're missing 4 to 5 million, or any new individual that's not the reference genome has 4 to 5 million single nucleotide variants between themselves and the reference human genome. Okay, add on top of that around 22 to 25,000 structural variants. These are insertions and deletions of at least 50 bases between an individual and the reference genome. And around 5 to 600,000 shorter variants kind of between that. Okay, so there's a lot of variation that all of us have that that's different relative to a reference genome. Okay, so what does that cause? Okay, if we're using one single reference genome to compare all of our sequence against, that's going to lead to potentially some problems in downstream analysis. Okay, obviously we're able to do some work, or a lot of work with a reference genome. If we're, say, able to confidently detect 5 million single nucleotide variants in an individual, we're able to do some good genetics with the reference that we have. But there are limitations, okay? For one, there's something called reference bias. So alignment algorithms, when you're detecting a variant, you're finding differences between the reads that you have and the reference genome. And those variances are heavily biased by things like alignment parameters, insertion and deletion scores, and mismatch scores. And because of that, you might have reads that are more likely to match the reference than the sample, even if the sample differs from the reference at a particular position. This plot here shows, on the left, this is showing essentially bias against reference alleles for indel alleles. And the denominator is doing a lot of work here. There's not as many sites out in the larger indel range, but as indels get larger, reads are less likely to be aligned in a manner that represents the sample's indel versus the reference's actual sequence content. And on the right, what's shown, this is work from Ben Langmead's lab, showing essentially the numbers of positions where there's new variants that shouldn't be essentially discovered in a sample that are because of what are called reference bias. Okay, so this is problems with a single linear reference that come up in very small scales. There's larger scales. If you're doing, say, short read sequencing and attempting to discover structural variation, we know that there's a very large gap between the number of variants that a sample has, 20 to 25,000, and the number of samples with, say, Illumina data that you can discover, which is around 5,000 structural variants per individual. Okay, and again, this is part reference bias, part difficulty in discovering structural variants with short read data. And then finally, kind of on an even larger scale, there's entirely missing structural haplotypes that occur across many individuals that are not going to be shown in a reference. So what's shown here is a region of a cluster of genes called amylase that this is, I believe, amylase 1. It's expressed in the mouth and it digests starch. Okay. Copy numbers in humans range from 2 all the way up to 8 copies that have been observed of amylase. And I believe the reference has 3 in GRCH38 for amylase 1. Okay. So if you have any of these extra 3 to 5 copies of amylase in your genome, any variation in your genome that you have in those extra copies is going to be missed when you rely on the single human genome reference to discover variation. Okay, so how do we solve that? So for a while, people have been talking about developing what's called a pangenome. And a pangenome is effectively just a collection of genomes that instead of saying we have one single reference genome, we're going to sequence many individuals, and the net group of all of those sequences is the pangenome. Okay, so I'm going to talk about 3 different aspects of the pangenome. So I'm going to talk about the design. So under either limited or even unlimited resources, what individuals would you like to include in a pangenome? We're going to talk about construction and then usage. Okay. And all pretty high level discussion of what's going on. Okay, so what genomes to include? Okay, so we don't have unlimited resources. We can't sequence everybody, but even if we did have unlimited resources, including every individual in the pangenome is going to scale computational methods for downstream analysis, maybe linearly, at best sublinearly, but an increase of computational cost for every additional genome that you add to the pangenome. So you might not want to include essentially every individual that you sequence in the pangenome. So the goal has been to essentially represent common human haplotypes, so allele frequencies of 1%. So these slides next few slides are courtesy of Karen Megan and Alice Popchoy. Looking at alleles with at least 1%, sample enough individuals that you have alleles of 1% frequency in the pangenome. Okay. For now, they've been leaning on samples that are in Coriell and are part of the 1,000 Genomes collection. So these are essentially low-hanging fruit. They're DNA that you can just go ahead and order, sequence, and add to the pangenome without any additional effort. But even with this, there's actually a bit of kind of fraught effort in having this definition. How do you define even 1% allele frequency? So we know the 1,000 genome sample collection isn't complete. We know there's populations missing from that. And depending how you sample individuals, even in 1,000 genomes, what turns out to be 1% allele frequency changes. Looking at the New York Times just recently had this article about kind of shifts in global populations. This was in the context of working populations, but so in 1990 we have these large population growths in East Asia, and over the next 50 years we're going to have this sort of shift of demographies into larger populations in continental Africa. So this, by its nature, if we did sequence every individual on the planet, this is going to change our measurement of allele frequencies and perhaps change the sort of definition of what individuals under that sort of allele frequency cutoff would we include in a pangenome graph. Okay. So the straightforward way to do this is essentially take 1,000 genomes individuals, separate out based on some diversity metric, looking at principal component analysis of SNPs in genomes, and essentially pick individuals that maximize the amount of variance between individuals that go into the pangenome graph. Okay. The sequencing that's been done for the human pangenome project has been moving along pretty steadily. Sorry, the years are kind of falling apart, but we're targeting 300 individuals by 2024, end of 2025. Okay, so this is 300 individuals, ends up being 600 haplotypes. It's actually pretty decent sampling of population diversity. And even with the individuals that we've sequenced so far, we're looking at a coverage of about 99.9% of common SNPs compared to the 1,000 genomes data set. But this PCA and maximizing the amount of variance between individuals going into the pangenome is actually only a moderately, well, kind of a tied component of sample selection in for building pangenomes, where you're not just maximizing variance and ordering from Coriell. There's actually a much larger component of essentially trying to link ethical, legal, and social implications for including individuals into the pangenome, where you're essentially not just trying to include an individual, but you're trying to get kind of societal inclusion of samples and populations that may have been historically underrepresented in genetic studies. So we know that for quite a while, a lot of genetic studies have been heavily biased towards European populations. And even since that's been kind of decreasing, the increase has been largely in increasing populations from Eastern Asia. And so we want to make sure that the pangenome not only reflects human diversity, but reflects essentially diversity of individuals who are participating in healthcare. Okay, so additions, categories for including individuals in the pangenome. So there's the diversity selections, but essentially going out and having a large amount of engagement with communities and making sure that people both are interested in participating in the Pangenome Consortium and that there's knowledge on how the genetic material and genetic information will be used. Okay, so next up, let's talk about building the pangenome. Okay, and building the pangenome essentially means collecting individuals, sequencing them, and assembling them. Pretty straightforward work. And one of the reasons why we haven't had a pangenome until just recently is because we've had this kind of massive leap in technological developments that have made assembling high-quality diploid genomes practical. Okay, so what's shown here is Haiyue Chang's HiFi-SM pipeline, where either this or a trio-based analysis was used to build assemblies in the pangenome. And essentially, HiFi reads, these are sequencing reads that are highly accurate, are built into a graph. They're combined with Hi-C data that I'll kind of show a little bit more information about in the next couple of slides, and use this to build assemblies. One of the key components of essentially modern assemblies, especially assemblies for the pangenome graph, is that we're all looking at diploid individuals, and so the assemblies have to be haplotype resolved. We have to be able to take the individual sequencing reads, partition out by haplotype, and then assemble two haplotypes separately. So the first essential ingredient for having this happen has been this development of long and highly accurate reads from PacBio. So this is a moderately old slide, but the average length that's shown here, it's a little less than 20 kb. This is a plot of Q20, I believe, so at least 99% accuracy reads. And so having these fairly long and fairly accurate reads has really made assembly a lot more straightforward, where essentially overlaps between reads can be filtered based on extremely high identity of overlaps, and it makes assembling highly contiguous genomes a pretty easy task, a relatively easy task compared to before. So the other sort of essential ingredient has been the use of a different sequencing data type that gives you long-range phasing information. And so that's either been trio information or Hi-C sequencing here. So Hi-C sequencing does this proximity-based ligation of sequences, and if you can sequence two parts of the genome that overlap heterozygous SNPs that are potentially from very distal parts of the genome, you can phase those two SNPs And you can use that for both phasing and scaffolding of the long read data. And so this is a view from just from a perspective paper that was published before the draft pangenome was out, just kind of talking about the different types of data that were combined to build pangenomes. But the main aspects are the fact that they had these highly accurate, moderately long read sequencing data and the scaffolding information from Hi-C. Okay. The result of this was a set of genomes that were assembled. What this plot shows here is effectively the quality of the assemblies, where it's showing the total amount of the genome that's covered by total length. And the darker black line here reflects the GRCH38 contiguity. And so anything that's kind of above this represents an assembly that was in the HPRC that's of higher quality than the 38 release of 38 on the human genome. And what's below, there's some samples that didn't have better assemblies than HE38, but by and large, the assemblies that the draft pangenome has are pretty high quality. Very, very contiguous, and it turns out things like multi-copy genes are assembled into this. Okay, so this was published somewhat recently. Something to always remember is if you're reading about anything in a publication, that means that you're reading about something that's already a year and a half, or a year to a year and a half old and potentially obsolete. Okay, so the set of high-quality genomes that we have here don't really reflect the state-of-the-art and the current process that's going on in the HPRC. And the sort of main new development of technology that's being folded into HPRC analysis is this new data, which is ultra-long read sequencing coming from Oxford nanopore. So these are the ability to sequence reads that are over 100 kb long, on many instances over a megabase long. And these sequences give you the ability to resolve extremely repetitive parts of the genome. So these ultra-long nanopore sequences combined with the really high quality, relatively long, but nowadays actually comparatively short, PacBio sequences and new assembly methods enable what's called telomere-to-telomere assembly of diploid data. So genomes, or at least chromosomes, that are, for the most part, contiguous from one end to the other end of the genome. And there's a large push in the HPRC consortium to try and aim for not quite telomere to telomere, but extremely contiguous assemblies. And so we have a lot of new information on, say, centromeric ribosomal DNA and other repetitive parts of the genome are essentially coming out into the newer release of the pangenome. Okay. I'm actually going faster than I need to be going, so I can slow down quite a bit. Okay, so using the pangenome. So kind of hand in hand with pangenome, a lot of people are currently mentioning the pangenome graph. Okay, how many people here have heard pangenome graphs sort of tossed around in one context or another? Okay, so so a pangenome graph is not the only way to represent a pangenome. Pangenome could simply be, or the pangenome is more or less defined as the collection of sequences that are assembled that one can build a graph on. You can also do things like leave them as sequences, and I'll mention some of our work later on that doesn't merge sequences into a graph. But the idea behind a pangenome graph is that it's essentially a very elegant way to represent a lot of fairly similar genomes that also have a certain degree of diversity, okay? Where each vertex will represent a sequence that's shared between a certain number of individuals, and edges connect sequences where there's going to be variation. Okay, so maybe there's going to be inserted sequences, single nucleotide polymorphisms, and kind of long gaps between sequences that represent deletions. Okay. So this representation is pretty elegant, right? Any new individual that we add, if one individual differs by a single SNP from all the other individuals that are in the graph, we don't have to add their whole sequence. We just add one extra vertex to the graph and a couple extra edges connecting them. Maybe you have to break one vertex into two. But it's a very good kind of compressed representation of many different related similar sequences. So how do we build that? Okay, so we're going to talk about 3 different approaches that you can use to build a pangenome graph. Okay, 2 are not ours, 1 are ours. One approach is essentially an iterative approach where you pick one genome that's going to kind of be a backbone that's sort of a standard reference to how your other genomes will be aligned, and then iteratively add genomes to this. And any time that you have, say, an insertion, you'll break one of your sequences into a couple different new vertices, add that new inserted vertex, and essentially keep mapping genomes onto this structure as in this sort of iterative approach. And this is Hung Lee's mini graph approach. Another approach pioneered by Eric Garrison has been to do this sort of all pairs alignment, where you have every pair of alignment of different genomes, and then essentially come up with a graph genome that reflects a simultaneous compaction of all of the different similarities that are defined by individual pairwise alignments. Okay. Both of these share one property, that the pangenome graph isn't the only part of the pangenome. A very critical part of a pangenome is essentially to retain what's called haplotype paths. So not only do you have the graph where each vertex is a sequence and each branching edge is a variant in the population, but you also keep track of all of the different paths of individuals that have been built into that graph. Okay. So why would you do this? So importantly, a lot of methods take advantage of linkage information. So if you have a SNP that's nearby a structural variant, it helps to really know that that SNP came from the same individual that had that structural variant. We'll talk about a method that takes a really good advantage of that later on. And then also, if you're doing read alignment, say you're taking a long read or a Illumina short read, and you're mapping it back to the pangenome graph, you might not want to allow any arbitrary path through that graph. You might want to only allow certain numbers of, say, switches between haplotype paths in the graph. So you're not kind of overfitting your sequence to the graph, and you're kind of simplifying the total number of different paths to explore when you're aligning a sequence to the graph. So a pangenome, so kind of the definitions we've seen so far, pangenome is not always a pangenome graph. It's a collection of sequences. But if you do have a pangenome graph, you really ought to keep around the haplotype paths that are in the graph. Okay, the third approach, it's something that we work on for an area of research that our lab specializes in, which is specifically looking at tandem repeat sequences. So this is important to us because tandem repeat sequences, it's a type of DNA that if we looked at these 20 to 25,000 structural variants between any two individuals, 60% of these are in tandem repeat sequences. And so we have an approach that builds what we call a repeat graph. It's a fancy name for a De Bruijn graph on tandem repeat sequences. And we have different ways of querying this type of a graph for essentially analyzing variation in these highly variable parts of DNA called variable number tandem repeats. Okay. Oh, wow, going through this way faster. The last time I was at CGSI, I went like 15 minutes over. So now I'm overcorrecting and going like 10 minutes early, which is good on a Friday afternoon. Okay, so applications of pangenomes. So the first thing, what do you have to do with a genome and a new sample? You have to do something with new reads and align to it. I mean, you can just study the characteristics of the pangenome, but that's not so interesting. We want to map new sequences to these pangenomes and find out what types of variation that weren't able to discover before. So Sirén, he's part of the group at UC Santa Cruz, has this method called Giraffe, and this is a pretty rapid tool to align at least short read sequences into a pangenome graph. Okay, in this approach, it leans heavily on knowledge of the haplotype paths in the graph. And the sort of killer app that's enabled by having a pangenome, at least with short-read data, is in genotyping structural variants. Okay, so I talked before about this huge gap between the number of variants known to exist in a particular sample, the number of variants that you can find with Illumina sequencing. There's a gap of about 17,000 variants that are missing with just calling variants with Illumina data. So it turns out that if in your pangenome, remember every branching path represents a variant, if you can have your read and find a path in that graph that covers a branch, then you can fairly confidently say that the sample that you have shares that variant in common. Okay. So depending how your reads thread through the graph, you can essentially genotype the different variants that are embedded within the topology of the pangenome graph. And so it turns out that you can get metrics by doing this sort of read threading with short read data into a pangenome graph that reflect about 90% of the variants that you would hope to find with, say, long read data by discovering variants in a graph. So leaning on the graph you're not discovering new variation, you're genotyping variation, but the number of variants that you're able to genotype at least get close to what you're able to discover with long read data. Okay, another very kind of closely related approach is a method called Pangene. Okay, so this is a similar to the Giraffe method. Instead of aligning reads, it uses k-mers and essentially looks for paths in the graph that are consistent with the k-mers in the sample. Okay, so again, depending on the paths that you're able to discover in the graph, you genotype all of the variants that overlap those particular paths. Okay, and so this histogram here is showing sort of a really key application, again, the genotyping of structural variants that are in the pangenome. What's shown here are variants that you can discover, or at least genotype in short read data in a pangenome graph, versus the number of variants that you would discover in a large data set where you're not relying on the pangenome graph, you're just using alignment and alignment and SV calling methods. Okay, and then the third application that we'll talk about is, again, this is something that we're doing in our lab. It's estimating VNTR composition using the repeat graph that I mentioned as kind of the third different way of building up a pangenome graph. And so what we can show is that if you take your pangenome and merge all of the VNTR sequences into a De Bruin graph, you can map reads into the De Bruin graph and essentially count how often reads map to different paths in the graph. And what that enables is the ability to actually find associations between repeat composition and various traits. So we had a study come out where we're able to do EQDL discovery on tandem repeats where we weren't necessarily looking for associations between length of a tandem repeat, but the actual composition of the tandem repeat and expression. Okay, so we finished this as a record time, actually. Okay, so let's talk about some future directions. So recently a paper came out as kind of a next step of the ENCODE project where essentially personalized genomes were used to annotate functional DNA. So this was a study only on 4 individuals, but they were sequenced with long read data. Haplotype resolved assemblies were produced on these individuals. And then essentially histone modification and chips were found, or peaks were found on the individual assemblies of these, personalized assemblies of these individuals. And so around 2 to 4% of reads were able to sort of map to the personalized parts of the genome. And what this means is that there's, okay, we're not rewriting genetics by having a pangenome. We're able to kind of, the improvements are indeed on the margins. We're able to have a decent number of additional functional elements that are defined when you rely on a personalized genome relative to kind of the one standard linear reference, okay? So if you get enough haplotypes built up into the pangenome, you can essentially piece together parts of any pangenome that will end up reflecting to a pretty close estimate what that individual's genome looks like. And then you can go ahead and, without sequencing that person specifically, you can do personalized genome inference on things like functional analysis or functional data or transcript level data. Another future direction is something that was my talk on Tuesday, which is allele and paralog specific copy number maps. So I mentioned very early on that the pangenome, or at least different individuals, have different copy numbers of certain genes in their genome. And what we wanted to do was develop a method that could detect which particular copies any individual has of a gene in their genome and whether or not they have additional copy number variants in that. What's shown here is an image from an old study in 2015 where Illumina read depth was used to estimate copy number of genes in genomes. This is Bob Hansaker's method, genome strip. And what they found is that certain haplotypes have what they call runaway expansions of gene copy number events. And so here are a bunch of different human haplotypes. And here, only in African individuals did this gene HPR go through these kind of runaway expansions. And so the details, I encourage you to go watch the talk on Tuesday, or from last Tuesday, but we have this method CTyper that essentially builds up databases on a per gene level of what all of the different alleles have and the different sequence variants any individual gene or paralog allele has, and then estimate both which alleles any individual has and whether or not there's copy number variants on that. What we can see is that there's different continental population or different distributions of these different alleles in continental populations from the 1,000 Genomes project. Okay. Thanks again to the Human Pangenome Reference Consortium, and thanks a lot to funding sources and the lab. So didn't touch too much on our work, but the slides that I did show from our lab were from Tony's work and Walfred's work. So since there is a little bit of leftover time, some open problems that I see in pangenome analysis. One thing we're working on, and I think is an open problem, is rapid sequence alignment to collections of genomes. So you have a large number of closely related individuals. You haven't yet turned them into a graph. I think there's some issues, especially around repetitive DNA, on determining how that graph should be built. So maybe just leave them as individual sequences. There's going to be a big push over the next few years on annotating the functional elements in the pangenome. So we have these beautiful graphs, wonderful sequences, and now we need to annotate both disease and functional information on top of that. So that sounds easy, sounds like we're just mapping sequences back to this, but there's a lot of complications. So for example, if you have a particular base in one genome that's could be aligned to a couple of different positions in another genome, how do you relate those positions between each other? Part of that's related to a difficulty in assigning gene orthology, especially when there's kind of rampant gene conversion that's going on between sequences. And then finally, I think we're having pangenomes that are reaching 600 individuals, so 1,200 haplotypes. 600 haplotypes. We need to be able to share those in a pretty highly compressed format. Okay. Any questions that you guys have?